students, let us revise the biological practical examination. Okay. See, in your practical examination, you will have the question paper. It is of like this, which is having 6 questions for which you have to answer. And the 7th one is Viva OC, 8th one is practical record. See, we have the question number 1 to 6. Instead of that, you have to write A, B, C, D, E, F. Say for the first question, that is A, you will have 4 options. A1, A2, A3 and A4. Then B, which is mitosis slide preparation. So both these two are the slide preparations. So this slide you have to show for the external examiner and this slide you have to show for the internal examiner. Then the C, we are having the biochemical test, that is the pH of water sample. You will have the 2 samples, C1 and C2, and you have to identify the pH values, and based on that you have to identify what is the nature of that water samples, whether it is acidic or basic. Then D, identify the specimen or slide or photograph, and you have to write 2 reasons. Then E, we are having 4 slides or photographs or specimens where you have to identify, then you have to comment on 2 reasons you have to write. For F, we are having some specimens or photographs where you have to identify and write down 2 characters. So, whenever you will be asked to enter into your practical examination, so your answer papers will be placed on a reverse fashion, so that first question, that is A question, you will have the by lot method. See, for example, just I want to tell you one typical example. See here I have 4 answer sheets. In one answer sheet I have written A1, then in the another answer sheet A2, then A3, A4. So these answer papers were placed in a reverse manner, and you have to pick any one of the answer sheet. Say for example this I have selected. For first question I have A2 question. So A2 means, so that is the preparation of the slide of polar tube growth on stigma. So that I have to prepare the slide, and whatever they have asked, that I have to write and focus the slide in the microscope, and we have to get the signature from the external examiner only. So anyhow, these are the aspects with respect to this answer sheet. See, dear students, as and when you get your answer paper, go to your place, write down the student number, then subject, register number, date, right? Then afterwards, as I already informed you in the regular practical classes, so fold the answer sheet in this fashion, 2 folds. So because of that, you will going to have 2 blocks in a page. So in the first page, first block is already filled with so many instructions. So second block, where you are having the space to write the required aspects for A1 slide. For example, A1 pollen grain germination percentage calculation. So you have to write down the tabular column and all those things. Okay? Then see here in the second page I am having 2 blocks. So write down here question number B, then C, then D, then E, then F. So if you write like this, so that you can answer all the required aspects in that suitable box only. So that will help the examiners so as to scan your answer paper within 1 or 2 seconds. Okay? Even though if you might have done any simple mistake, if you written in this fashion systematically, so they can glance your paper just within a second, and they can recognize the mistakes, and they can give an option to rewrite the aspects or correction. Okay? So this is how you have to maintain your answer paper. See, this is the rack where the A1 question pertaining materials are kept. So A1, A2, A3, A4. The flowers, stains, or the required solutions, all the requirements that are required to perform A question slides that have been placed in this part, in this rack. And here in the second rack, so B, that is mitosis, onion root tips you require, then acetocarmine stain you require, all those things were kept here. Then C, pH of water samples. So 2 water samples will be kept here, C1 and C2. In between these two, we are going to keep the required pH universal indicator solution. Okay, so the students have to come to this place, and whatever you require, your requirements to prepare to conduct the respective experiment, so you can collect these materials and go to your place and perform the respective experiments. Then similarly, so as far as the D, so D is pertaining to spottings, identification and 2 characters. So this is the place where the D specimen or photograph or the slide is placed. Then coming to the E, so E also the slide or photograph or specimen. So here we will going to keep. Then similarly F, last. So there also we are having the specimens, so or photographs. Here we will going to have. Whenever you are coming to these places to identify these spottings, you should come with your answer paper, then pencil. So you are not supposed to write anything on the question paper, only you have to write the identification name with your pencil only. Okay? Then go back to your place, original place, and then you can write whatever you want in a neat fashion. Okay? So let's see the first question. So first question we are having A. This is the A. Under this A we have 4 choices, 4 options. A1. A1 is, this is the experiment or this is the slide preparation of the pollen grain germination percentage calculation. For this we require the flower Vinca rosea, then sucrose solution, isn't it? So these are your requirements. So these things will be kept in the track as already shown, explained previous, just now within 5 minutes back I explained, isn't it? So in that rack we are going to keep these requirements at A1 place. Then by taking these requirements, and you have to prepare the slide by using the cover slip and everything. Keep it in your mind, you have to use only the sucrose solution to prepare the slide. Then A2. So this is the second slide preparation with respect to pollen tube growth on stigma. So here also we require Vinca rosea flower, then cotton blue stain to prepare the slide. So after preparing the slide, you have to draw the label diagram for this question, isn't it? Pollen tube growth on stigma, you have to write the label diagram. For A1, what we have? Tabular column and calculation. We have to identify the percentage of the pollen grain germination. For this, you have to write the label diagram of the pollen tube growth on stigma. Then A3, so this is the slide preparation of the transverse section of ovary. So for this slide preparation we require hibiscus flower. Then in that flower we have to take out the ovary and take the transverse section of the ovary, and those transverse sections were placed in water. Then among the 4 or 5 sections you have to select a thin, clear, complete section and prepare a slide by using water and place a cover slip. Then you have to focus that slide only this dissection microscope, most important. For this, what we are having the question, so after preparing the slide, the question is how many locules are present in an ovary? There are 5 locules. Then what is the type of the placentation? Axial placentation. So these things you have to write. Then we are having the A4. So this experiment is the nuclear staining experiment. For this we require onion pieces, then acetocarmine we have to use. It is commonly called as a nuclear stain. So here, by using this nuclear stain, we are going to prepare a slide. For after the preparation of this slide, So before showing this slide to external examiner, you have to write 2 points. So for slide preparation, 3 marks. For 2 marks, what you have to write? Shape of the cell. The shape of the cell is found to be rectangular. Then number of nuclei in a cell. So as it is a plant cell, only one nucleus is found and it is at the periphery. Why? Because of the presence of a large vacuole. So these are the aspects. So 3 marks for slide preparation and 2 marks for writing 2 points. Here 2 marks, 2 points. Here you have to write down the label diagram. Then here you have to put the tabular column and you have to find out the percentage of the pollen grain germination. So then coming to the second question, that is B. So B is with respect to mitosis. So here for this, here also the slide preparation, second slide preparation. Each student will have to prepare 2 slides. One, the slide which was prepared is to be shown to the external examiner. This is the slide which is prepared by all the students. You should show this slide to the internal examiner. So in order to prepare this mitosis slide, we require onion root tips. So these are the onion root tips. So in your table also, you will have the watch glass. You have to come with forceps and watch glass, and you have to take out 1 or 2 onion root tip, go to your place. Then, so these are the things that are required to prepare the slide for mitosis. So this is the 1 normal HCl, then acetocarmine, which is a nuclear stain, and this is 45 glacial acetic acid or 45% acetic acid. So here what you have to do is onion root tip. For this you have to add 1 or 2 drops of 1 normal HCl. Then you have to heat it for 1 or 2 seconds. Then this onion root tip is taken and it is placed on the slide, and you have to add 1 or 2 drops of acetocarmine stain. Then leave for 30 minutes. After that, remove the excess stain. Then this slide is prepared by putting 2 or 3 drops of this acetic acid, 45% acetic acid. Put a cover slip and tap that, okay, so as to squash that material. Then this slide should be focused in a compound microscope, and what you have to do is, for the preparation of this slide, you will get 3 marks. Then you have to write the label diagram of any one stage of mitosis. You will have an option, either you can write prophase or metaphase, anaphase or telophase. So better to write prophase stage. Okay, don't write telophase or anaphase. Okay, so keep it in your mind. This slide should be shown only to the internal examiner, and you should get the signature for the label diagram which you have written in your answer paper in the second page of first block. Okay, then coming to the third question, that is C, where you have to write pH of water samples. So in that rack, we are going to keep like this: 2 water samples. One is C1, the other is C2. In between these two, we are going to keep a pH universal indicator. On this bottle only, you will have the pH scale, where each color has the pH value. So see here, 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Anyhow, you are having the pH scale which range from 0 to 14, isn't it? But here, whatever the samples which we are going to give for you people during the examination, the pH value comes in between 0 to 10. That's why in this pH scale, we are having the value ranges from 0 to 10. So here what you have to do, you have to take 2 test tubes. In one test tube, you have to add 1 or 2 drops of the C1 sample. In the another test tube, add 1 or 2 drops of C2 sample. For both these test tubes, you have to add 2 drops of pH universal indicator solution. Then afterwards, shake properly. Then compare the color of both these test tubes with this pH scale. So whatever the values we are having on this bottle, that will be the pH value of that respective sample. Okay, so definitely one will be acidic and one will be basic. Okay, ಎರಡು ಯಾವುದೇ ಕಾರಣಕ್ಕೂ acidic ಇರೋದಿಲ್ಲ, ಎರಡು basic ಇರೋದಿಲ್ಲ, and also you won't get even the neutral solution. Okay, so one acidic and one basic. So after conducting this test, you have to draw a tabular column where you have to write down the C1 sample based on the pH color. You are going to write the pH values. Based on that, you are going to identify what type of the sample it is. If it is the value is less than 6.9, then it will be considered as acidic. Then if the pH value is more than 7.1, then it will be considered as basic. So after writing all these things in the tabular column, compulsorily the student has to write the report about this experiment. That is, you have to say in your own sentence what is C1. C1, if it is acidic, the given C1 sample is acidic and the given C2 sample is basic. Okay, so these are the aspects with respect to C experiment. Then after getting the signature in your answer paper, either from the external examiner or internal examiner, then only you will have the permission to wash your test tubes. Till the signature, you are not supposed to wash your test tubes. Okay? Anyhow, move on to the D, that is fourth question. Fourth question. So that fourth question includes. So this is the D. D we have. So here identify the photograph or slides. We have two photographs and two slides. So one is, this is the VS of ovary showing female gametophyte. ಅದು ಏನು ಬರೀಬೇಕು? The given photograph D is identified as VS of ovary showing female gametophyte. ಸುಮ್ಮನೆ ಸಿಂಪಲ್ ಆಗಿ ಫೀಮೇಲ್ gametophyte ಅಂತ ಬರೆಯೋದಲ್ಲ. So you should write VS of ovary showing female gametophyte. So you have to write any two characters. Then this is the another photograph. It is VS, the given photograph D is identified as VS of mammalian blastula. So blastula is a single layered structure. There is a presence of blastocell. Then the third one, we have the slide. The given slide D is identified as TS of mammalian ovary. So for this D, we have two slides. Okay, this is the slide which is called as TS of mammalian ovary. See here. I want to give one easy tip. The slide is, the stained part is very small. Okay, this is the one tip. Then the another tip is, this ovary slide is kept under dissection microscope. Okay, then we have another slide under this D. It is TS of mammalian testes. See here, this slide, the stained part is quite larger in size. Okay, and then so this slide is kept under dissection microscope. That is compound microscope. Sorry, it is kept under compound microscope. Clear? TS of mammalian ovary slide is kept under dissection microscope. TS of mammalian testes is kept under compound microscope. Say, let's move on to the next question, that is E. So for this E, in the regular practical classes, we have shown Entamoeba histolytica photograph, then Plasmodium vivax photographs, Ascaris lumbricoid specimen, then Trichophyton rubrum. This is the photograph. So E question, identify the name of the pathogen and name the disease it causes. See, along with this Entamoeba histolytica, this photograph, we are having a slide.
In the practical examination, 90% the external examiner will prefer the slide. Okay. Rarely they may prefer the photograph. Anyhow, all the students should aware about both the photograph and as well as slide in order to identify the entamoeba histolytica. It causes amoebic dysentery. Then Plasmodium vivax. Here also photograph and also a permanent slide. 90% of the examiner will prefer the slide. So in rare case you may get this photograph. Anyhow, you have to observe both the photograph and as well as the slide. So Plasmodium vivax, it causes malaria. Then Ascaris, it is an endoparasite of man. It causes the Ascariasis disease. Okay, this is the live specimen, preserved specimen. Okay, Ascarias lumbricoides. Then Trichophyton rubrum, photograph, it is a fungus. It causes a disease called ringworm disease. 4 pathogens and the disease it causes. Then move on to the next question, that is F, where so in this F we are having 8 options or 8 specimens. So out of these 8 you may get any one. You have to identify the specimen or photograph, okay, and you have to comment on that particular specimen or photograph. Say this is the Pistia. Pistia is an hydrophytic plant, the plant which is found in an aquatic habitat or water body. Here we are having the photograph Pistia. So even you may get the live specimen also. Okay. So ಈಗ ಇಲ್ಲಿ ನಮ್ಮಲ್ಲಿ ಅವೈಲಬಿಲಿಟಿ ಇಲ್ಲ, ಎಕ್ಸಾಮಿನೇಷನ್ಗೆ ತಂದಿಟ್ಟರೆ ಅಕಸ್ಮಾತು you should be able to identify that material or the specimen as Pistia. Then Eichhornia, it is also an hydrophyte. Eichhornia, this is the live specimen. So this you can differentiate from the Pistia as it contains the pulvinated leaf bases or the swollen leaf bases, so which contain air pockets. So presence of parenchyma in the hydrophytic plants is the most important striking feature of hydrophyte. So Pistia, Eichhornia, either photograph or the live specimen. Then the xerophyte, xerophyte opuntia. So this is the photograph, and this plant you might have seen, it's a xerophytic plant which grow in water scarcity habitats. So where the leaves are reduced into spines and the stem is modified into a leaf-like structure called phylloclade.
Identification, fish. So fish. So here the body is spindle shaped, body is covered by dermal scales, then it has the fins as the locomotory organs. Then respiratory organs are gills. Then camel, it is an example for xeric animal. Presence of hump, then presence of foot pad, like this you have to write. Then homologous organs. Homologous organs. What are homologous organs? So origin is same, but the structure is different. So origin is same, structure is different. Because of the structural difference, they perform different functions. They are called as homologous organs. See here, it is the say same origin, structure and functions are different. So here, this is the identification. Thorns of citrus or lemon, tendril of cucurbita plant. Origin is same, that is in both the cases axillary bud is modified here into spines, here into tendrils. Origin is same, axillary bud. Structure, here it is spine, here it is tendril. Different structure. Because of the difference in their structure, they perform different functions. So here the spine used for protection, this tendril used for support or climbing. So these are the available photographs. This is the one set. This is the another. It is a specimen, preserved material, preserved specimen material. Okay. So homologous organs in plants. See here, homologous organs in animals. What is this? Photograph is identified as forelimb of bat and forelimb of man. So origin remains same, but in case of the man, the forelimb is used for different purposes. Whereas in case of the bat, the forelimb is modified, it is used for flying. Okay, so performing different functions because of the difference in their structure. Even though they are having same origin, different structure with different functions are called homologous organs. Homologous organs in animals, homologous organs in plants. Then come to the analogous organs. What are analogous organs? So here origin is different, whereas function remains same. Okay? So such organs are called analogous organs. Example, analogous organs in case of the plants. So this is the carrot, which is an underground modified root. It is the potato. It is an underground modified stem. Both these are meant for storage of food material. Different in origin, performing same function. See, this is wings of birds and insects. So origin is different. Whereas, so in case of the birds, forelimb is modified into wings. In case of the insects, these wings are a dead material. So both of them are performing the same function, that is helps for flying. Okay, so these are the aspects, students, as far as regarding the practical examination.